Okay, hello all. I'm sorry that I couldn't hold a live review class, but things just did not align this week. And I wanted to make sure that everyone had the opportunity to watch the review class or participate in the review class. And so I figured recording it would be the next best option. So I'm going to just start, as usual, with kind of a rundown of what you can expect on the exam, kind of the breakdown of the chapters, and then I will go through each chapter's review slide set, and that will be it. So let me take a look through the exam. So as usual, there's a couple of different parts. Part 1, multiple choice questions. There are 25 multiple choice questions. There are, let's see, 1, 2, 3. So there are 8 questions from chapter 12, multiple choice questions from chapter 12. And just to give you a breakdown, there are 3 that are on monohybrid crosses. So hopefully that makes sense to you. There is a question that involves the term test cross. So you want to make sure you know what that means. So when you see the word test cross, the words test cross, you should know that you're going to cross whatever organism in question with an organism that is homozygous recessive. So that test cross means crossing with a homozygous recessive organism. Okay, there is a dihybrid multiple choice question. There is a blood type multiple choice question. There is a multiple choice question on epistasis. And there are 2 multiple choice questions on X linkage. So again, that's 8 from chapter 12. If I just go over to the matching questions, which is part 2, it says part 3 on here. Hopefully I changed that on your copy. For matching, there is 1 matching question with 1, 2, 3, 4, 5, 6, 7, 8, with 8 parts to it. Again, this one's from chapter 12, and it's all of the genetics terms. So you want to make sure you know things like phenotype versus genotype, dominant versus recessive. You should know what the term locus means. You should know homozygous versus heterozygous. You want to make sure you know what the term allele means. Okay, so a bunch of just basic definitions from chapter 12. And then if I go to the short answer, so this would be part 3, there is 1 short answer or free response question from chapter 12. It has 2 parts. The first part is just a basic dihybrid. So you want to make sure you know how to set up the Punnett square and solve for that. Okay. And then the second is on blood type. So you want to make sure, again, you know how to set up the Punnett square. And the blood type question, remember that AB blood type is an example of codominance. And so you want to make sure that you understand what codominance is. Okay, so that's it for chapter 12. And again, I'll go through these review questions with you in just a second. From chapter 14, which remember was all about DNA structure, function, replication. So chapter 14, there are 1, 2, 3, 4, 5, 6, 7. Yep, there's 8 multiple choice questions. A couple of those questions, a lot of those questions are on DNA structure. So, you should know the bases in DNA and RNA. You should know, what holds the 2 strands of DNA together. You should know the term nucleotide. You should know about DNA replication. You should know all the enzymes. So helicase, DNA polymerase, primase, all those. There's another question on structure. So remember the measurements. We went over some measurements of DNA. We talked about the width, the distance between 2 consecutive bases, 1 complete turn, or 10 base pairs. So you should know those numbers. You should know that DNA replication is semi-conservative, and you should know what that means. So you want to make sure that you can, define that. There's a multiple choice question on that, but there's also a short answer on that, which I'll get to in just a second. So again, structure and function of DNA is important. Let me go to the matching. So the matching question is all about DNA replication. So it's all the enzymes. So again, DNA helicase, DNA polymerase, primase, topoisomerase, ligase, all those enzymes. You got to make sure you know what they are. And other things involved in DNA replication. So single-stranded binding proteins. You should know lagging versus leading strand. You should know what Okazaki fragments are. Okay, so again, lots of those. And then let me go to the short answer questions. So there's one where I ask you to explain DNA replication. So here is where you have to know that it happens in a semi-conservative fashion, and you have to be able to explain what that means. Okay? You have to know old versus new, all that kind of stuff. I ask you to talk about each enzyme involved, and I include a list of enzymes. And if you're smart, the matching question, if you got those right, go back and look at that. You can pull information from earlier steps in the exam. The definitions of all these enzymes are actually in the exam for you. So you want to make sure you talk about each enzyme and talk about them in the right order. Make sure you go in the order that they operate, that they work. Helicase is first. It unwinds and it unzips. Primase comes in, lays down a short stretch of primer because DNA polymerase requires a free 3' end. DNA polymerase comes in, it does its job, and so on and so forth. So you want to make sure that you go through all of them. And in this question, I do ask you to explain the difference between the leading and lagging strand, and make sure that you make reference to Okazaki fragments. And I ask you for a diagram. And so you want to make sure that you can explain, as well as make a nice little sketch, a labeled diagram, showing, where these enzymes work, which strand is the leading and lagging strand. You're going to have to make sure that you label 5' and 3'. Okay, all of those things, are going to be important here. And then the last question, the last short answer question, I'll touch on that now. It's really from chapters 14 and 15, so I'll talk about it now. What I ask you in this last short answer question is to really dive into the structure of DNA. So I want you to talk about what a nucleotide is. What's the backbone of the molecule? What does that mean that it's the backbone of the molecule? What's on the inside of the molecule? How are those two strands held together? Talk about how it's double-stranded, how it's a helical structure. Talk about base pairing rules. And then you want to compare DNA and RNA. So how are they similar? How are they different? And I ask you to highlight at least 3 differences. So again, here you might want to talk about the sugar, right? So the sugar in DNA is deoxyribose, RNA it's ribose. You might talk about the bases, which bases are present in DNA versus RNA. You might talk about overall structure. DNA is double-stranded, RNA is single-stranded. DNA is this big long molecule made up of many genes. RNA is short, made up of only one or a few. Okay, so you want to hit all of those. And then let me just go back to the multiple choice. So from chapter 15, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 multiple choice questions. They're on transcription and translation. Okay, so you want to make sure that you understand overall what those 2 processes are. You should know what a codon is, what an anti-codon is. You should know what RNA polymerase is. You should know basic differences between prokaryotic and eukaryotic transcription and translation. Basically, how does the nucleus come into play? Where does transcription happen in eukaryotes versus prokaryotes in the nucleus? You should also know that in eukaryotes there has to be RNA processing. And so you want to make sure that you know what RNA processing is. You should understand the 3 different types of RNA. There's a question just on one of them, but again, you should know actually 2 of them. There's a question on mRNA, and there's a question on tRNA. So you want to make sure you know those 2. You want to make sure you know what the ribosome is, and you should know that after protein is made, it gets folded up. Okay, and there is one matching question. There's only 4 parts to it. Again, same things that I just talked about are in here as well. Okay. So there are parts of, all the chapters, 12, 14, and 15 that I don't touch on. So for example, there's nothing on mutations. There aren't any specifics on the steps of transcription or translation. So don't spend a lot of time, going over initiation, elongation, and termination. There's no specifics on that. I don't ask you to sketch them or describe them in a short answer question. Okay, so hopefully that helped. Let me jump into chapter 12 here. Hopefully this works. Okay, so I'm on the review slides for chapter 12. Let's do number 1. It says, imagine you are performing a cross. Okay, so this is genetics involving seed color and garden pea plants. Sorry if my dog is breathing loud. It's about to storm and he's anxious. So if you can hear that, that's him. Okay, so imagine you're performing a cross involving seed color and garden pea plants. What offspring would you expect if you cross true breeding parents with green seeds and yellow seeds? Yellow seed color is dominant over green. Okay, so take a minute and try to solve this, and I will too. So yellow is dominant. I'm going to use the letter Y. So big Y, or big Y, little I is going to give me yellow, and green is going to be little y, little y. And so the question says that I'm crossing true breeding plants. So what does that tell me? That term true breeding or pure means that they are homozygous. And so the yellow must be homozygous dominant, big Y, and I'm going to cross that with a homozygous green, little y. And so the answer here would be B. 100%, so the offspring, 100% of them should be heterozygous, big Y, little y, and their phenotype would be yellow because dominant's yellow. Yellow is dominant over green. So B would be the correct answer. Okay. Next question is just a definition. Again, these are on there as well. The observable traits expressed by an organism are described as its, hopefully you answered phenotype, so that would be A. A recessive trait will be observed in individuals that are blank for that trait. So the only way to see the recessive phenotype is if the organism is homozygous recessive, so that would be C. Okay, here's a question on the law of segregation. The law of segregation states that, let's read these real quick. A lot of these sound kind of similar, but C is the correct answer. So alleles for a given trait separate or segregate during gamete formation and end up in different gametes. Okay, there's Mendel's first law. Okay, next one, it says, if a pea plant, here we go, so we got a genetics problem, I got to write these out. If a pea plant is heterozygous for height, what percentage of the offspring will be tall? So I'm guessing tall is dominant to short. I didn't put that in here, but it is. If we cross with a homozygous tall plant. And so that tells you right there that it's dominant. Okay, so it's going to be big T, little t being crossed with big T, big T. And so if you need to do the Punnett square, you can. But hopefully just by looking at this, you know that 100% of them, let's see, what percentage of offspring will be tall? Yeah, all of the offspring will be tall. Okay, half will be homozygous dominant and half will be heterozygous, but they will all be tall. So D. Next question, the Punnett square, it's a diagram used to determine, and so we use it for both. We can get phenotypic and genotypic ratios. I usually ask for the phenotypic ratio, but you also have the genotypic. And so C would be the most correct answer. Okay, the next question says, which of the following represents a test cross? Okay, D. So this is going to be crossing an individual that has the dominant phenotype. In this case, it's a heterozygote. It could be a homozygous dominant with a homozygous recessive. So D would be the correct answer there. So again, whenever you see the term test cross, which you will see on the exam, you're thinking homozygous recessive. Okay, so here we go, another one. If two heterozygotes, so big R, little r, times big R, little r, are crossed for this trait, whatever the R trait is, what percentage of their offspring will be homozygous recessive? So let me give you a minute to solve that. Okay, so hopefully you did your Punnett square, and 25% of the offspring are expected to be homozygous recessive. That's the prediction. Okay, Mendel's second law, the law of independent assortment, relates to the inheritance of, let me give you a minute to answer this one. So hopefully you got B. So this is going to deal with genes, different genes. So we're studying two genes at the same time, so dihybrid. And those two genes have to be located on different chromosomes to follow this rule. If they're on the same chromosome, those are called linked genes, and they won't follow this law. Okay, so B is the correct answer here. Okay, another application question, which again, the exam has a lot of these on them. So here we go. In peas, yellow's dominant to green again. Okay, same thing. And smooth is dominant to wrinkled. So now we have two genes. This is a dihybrid. So I got to write this out. So yellow is going to be homozygous recessive. So again, I'm using Y. You can use any letter you want. And the second gene is going to be seed shape. And so we've got smooth. Got wrinkled. So it looks like smooth is dominant. So I'm going to use S. Wrinkled is going to be homozygous recessive. Okay, so it says a plant that is homozygous dominant for both traits, so big Y, big S, is crossed with a plant that is homozygous recessive for both traits. So little y, little s, okay? It says what type of seeds will be produced in what ratio. So again, write that out. So what you should see is that all of the offspring will be big Y, big S, little s, or we could say heterozygous for both genes. And they will be yellow and smooth. Correct? Yeah. And so C is the right answer. So all seeds will be heterozygous, and they will have the dominant phenotype for both genes. So yellow and smooth. Okay, next question. It says, what is the procedure called that is used to determine the genotype of an organism with a dominant phenotype? What type of organism would you use to perform this? So this is going to be a test cross. Oh, let me give you a second to answer. Okay, so hopefully you answered D. So this is going to be called the test cross. And whenever you see that term, you know you're using a homozygous recessive organism. Okay, my poor pup. Okay, next question. It says, if black and white true breeding mice are mated and the result is all gray offspring, what inheritance pattern would this be indicative of? Okay, so we've got black and white. Okay, so we got black and we got a white mouse, and they're mated and they end up with all gray. Okay, so got black, we got white, we cross them and we end up with gray offspring. That's an intermediate phenotype. So off the bat, you should tell me this is probably an example of D, incomplete dominance, right? So just like our 4 o'clock flowers, you mate red and white and you end up with pink. Same idea here. And sometimes students will say, well, why isn't this codominance? And so let me tell you what this would look like if it was codominance. So if I use the same scenario, if I said I had black mice and I bred them with white mice and I ended up with white mice with black spots, that would be codominance. You see both phenotypes in the offspring, But this is not that. Like the result here are gray. And so that's an intermediate. It's in between black and white. So again, that's an example of incomplete dominance. Okay, here's the next question. I've got to get the back sheet of my paper here. It says a farmer raises black and white chickens. Okay, so we got black chickens and we've got white chickens, and he's breeding them. And he says, to his surprise, when the first generation of eggs hatch, all the chickens are black with white speckles throughout their feathers. So again, this is codominance. And then it continues to say, what should the farmer expect when eggs are laid after breeding the speckled hens? Okay, so again, you want to designate this. It's not true dominant recessive, it's codominance. So I'm going to use the letter B, and I'm going to say that black is B1, B1, and white I'm going to say is B2, B2, and speckled is B1, B2. So again, you want to make sure that you designate it that way. Don't use a capital B and lowercase b. And so then you have to do the cross. So if you cross two speckles, so B1, B2 times B1, B2. And again, you might already know the answer to this just by looking at this. But if you need to do the Punnett square, you can. I'll give you a second to do that. So hopefully you've gotten, it should be a 1 to 2 to 1 ratio. And so there should be 25% black, 25% white, and 50% should be speckled. And so let's see, what answer is that? That's C. Okay. Okay. I don't think, let me just look through. There's a blood type question, which again, that is a codominant question. But other than that, there isn't any other codominant or incomplete dominant, although they're very interesting. Okay. There is a question on epistasis, so let's, oh, I just gave you the answer. Let's read through it though, because I want to make sure you understand this. It says in Labrador Retrievers, this is the same example that I gave you in the PowerPoint, one of the examples. It says Labrador Retriever fur color is controlled by two genes. We've got the E gene and the B gene. So it's a dihybrid, two genes controlling one trait. Not only is it a dihybrid, but again, you have two genes, one trait. So it's a polygenic trait, two genes, one trait, instead of two genes, two traits, two genes, one trait. So that's called a polygenic trait. But there's a special relationship here. It says any dog that is little e, so homozygous recessive for the E gene, develops into a yellow lab. Okay? While any dog that is big B dash, big E dash, become black labs, and little b, big E dash, become chocolate labs. And so you have to kind of understand this example. And again, I went over it in a previous, in the chapter 12, part something, lecture, I think part 3 maybe. Notice here that if there are two recessive E alleles, you don't get color deposited. So yellow labs are kind of like the albino, right? There's no color deposition in the fur. Anytime you have a big E, you get color deposition. So if you have a big E and a big B, you get a black dog. If you have a big E and a little b, you get a brown dog, a chocolate lab. The only way to get yellow is if it's little e, little e. It doesn't matter what's there for the B gene. It could be big B, big B, little b, or little b, little b. If there's not a dominant E, you don't get color deposited. Okay, so that is an example of epistasis because the E gene controls the expression of the B gene. It all depends on what's there for the E gene. Without a dominant E, you can't get color. Okay, so again, this is an example of epistasis, which is A. Okay, which of the following is an example of a sex-linked trait in humans? And so there are a couple of X-linked or sex-linked examples, and actually the one listed here is on here. So hemophilia is an example. And you'll have to solve that. There's more to the question than that, but It's good to know that hemophilia is X-linked. That is written in the question. I don't assume that you memorize that. The rest of these are not sex-linked. Okay, here's a blood type question. It says a child with blood type O. Okay, so kid, O, and I'm going to write the genotype. You know I like to write out everything. The mother is blood type A. So mom, she's A. And she can either be IA or IA little I. Okay. Which of the following could not be the biological father? Okay, so you got to go through each one of these. So could she have mated with somebody else with type A blood and produced a type O child? Yeah, absolutely. If her and the man were both IA little I, then yes, they could have a child that is type O little I little I. There would be a 25% chance of that. A man with type B blood, yeah, same thing. As long as he's IB little I, then yeah, still 25% of their offspring will be O little I little I. They'd have to both be, IA little I, and the male would have to be IB little I. Okay, C says a man with the blood type AB. Okay, so he would have to be IA, IB. Is that possible? Take a minute to think about that. That's not possible. Okay, so again, the child is type O, which is little I little I. Okay, so the answer, so C would be the answer. A man with blood type AB, it's not possible, right? So he's IA, IB, and so there's no way that he doesn't have a little I allele to contribute to the child, right? And so the child who's type O has to get the little I allele, one from each parent, okay? And D, yeah, if the father is blood type O and the mother is IA, little I, then yeah, they could have a type O child. So the only answer that makes sense there is C. Okay. Next question says traits that are controlled by multiple genes showing continuous variation are referred to as polygenic traits. Okay. So that would be A. There's no questions on those on the exam. Okay, so this one says X-linked recessive traits in humans or in the fruit fly, so this is Drosophila, or in any organism. Which one of these would be correct? So hopefully you put A. They're observed more often in males than females. And the reason why is because males are hemizygous, so they only inherit one copy of the X-linked trait, and therefore they're more likely to inherit a recessive trait. And sometimes those recessive traits are deleterious, bad, okay? So for example, males are more likely to have red-green color blindness, which is an X-linked recessive trait. Males are more likely to have hemophilia, again, X-linked recessive trait. Okay. All right, next one. It says hemophilia is an X-linked recessive disorder. And so here's a problem for you. And again, you want to be able to solve these. There are a couple of X-linkage questions on the exam. It says a woman who is a carrier. So that term carrier means she's normal. So she does not have hemophilia, but she's a carrier for it. So one of her X chromosomes has the normal allele, the dominant allele, and then her other X chromosome has the recessive, the bad allele. So she would be X big H, X little h. She's a carrier, okay? She has children with a man who does not have hemophilia. So he would be X big H, Y. So he's hemizygous. He only has one copy of this gene, and it's a dominant form of it because he doesn't have hemophilia. Okay, so do the cross. And it says, what's the probability or the percentage here that a son will have hemophilia? So we don't care about the daughters. Well, we care about the daughters, but they're not in the question. So you got to set up your Punnett square. And so X big H, X big H, X big H, X little h. So for female children, half of them will be homozygous dominant and half of them will be carriers. For the male children, half of them will be normal and half of them will have hemophilia. So the probability of having a son that will have hemophilia. Yeah, so here you would just look at the sons, and so half of the sons are expected to have hemophilia. Okay, so the way this is worded, you got to make sure you answer it right. So what is the probability that a son will have hemophilia? So you just have to look at the male children, and out of them, how many will have hemophilia? And they would be half. So you got to be kind of careful on this question. So that would be C. The question. Yeah, so there is a question just like this. So you want to make sure that you're looking at just the sons. So some people might say, well, 25% of the offspring will be males that have it, but that's not what the question asks. It asks you, out of the sons, how many are expected to have it. So be careful there. Okay, color blindness. Here's another one. So color blindness is X-linked recessive. Okay, we have a colorblind woman, so she must be X little c to be colorblind. She's not just a carrier, she's got to have both recessive alleles. She mates with a man who has normal vision, so X big CY. And it says, what will be true of all of their daughters? Okay, so let me do this. And again, you may not have to actually do the Punnett square, but if you do, my OCD makes me do them all the time. I have to double check everything. And so the answer here, all of their daughters, they will be heterozygous carriers. They'll have normal vision. So let's see which one of them. Yeah, so B. None will be colorblind. All will be carriers. Okay. Oh, and that is the end of the chapter 12. And I think I'm going to cut the video here and do a new video for 14. So please click on the next one.